Welcome to our webinar. We thank you so much for joining us today. We're just going to give the audience a couple of minutes to join us, and then we will get right to our program. We appreciate you being here today. My name is Abby Ingber, and it is my pleasure to welcome you to today's webinar, Unraveling the Genetics of Borderline Personality Disorder. I'm the Executive Director of the National Education Alliance for Borderline Personality Disorder. We are a nonprofit organization that provides education, raises public awareness and understanding, decreases stigma, promotes research, and enhances the quality of life for those affected by borderline personality disorder and related problems, including chronic emotion dysregulation. We have the largest virtual resource library online with information related to borderline personality disorder. We bring together clinicians, researchers, family members, and people with lived experience at conferences around the world. Our signature program is Family Connections, which is a 12-week course for caregivers of individuals with BPD to learn psychoeducation skills and receive support. We also have a course for parents in distress due to their child's previous suicide attempts, self-harm, or other dangerous behaviors. This is our Managing Suicidality and Trauma Recovery course. Please feel free to check out our website at www.neabpd.org. Before we begin, we just have a couple of housekeeping items to cover. This program is being recorded and will be uploaded to our website. Your video has been disabled and you are on mute. We ask that you submit your questions via the Q&A, not the chat. The presenter will answer as many questions as time permits. We are going to ask that you keep your questions specific to the presentation topic, as this is not a forum to ask for advice for a personal situation you may be experiencing. The opinions expressed today are those of the speaker. They do not necessarily represent the views of NEABPD. This webinar is not intended to be political or to be therapy or a substitute for therapy. As an education-based nonprofit, this webinar is meant to be educational. We do not intend to say anything that will be triggering, and we ask that you exercise self-care. If you do find something that is activating, please sign off immediately and tend to your needs. 988 is available in the United States 24/7 as a resource. Please reach out to your mental health provider or support system if needed. We thank you for being part of the solution in transforming so many lives. We offer webinars like these for free, knowing there's a huge need. We thank you for your support in continuing to offer fantastic programs like this one today. It's my pleasure to introduce Dr. Fabian Streit. He is a researcher at the Hector Institute for Artificial Intelligence in Psychiatry at the Central Institute of Mental Health in Mannheim, Germany. His work focuses on understanding how genes and life experiences shape mental health, especially in conditions like borderline personality disorder. He leads international collaborations exploring the genetics of personality and emotional well-being and is part of several large research efforts aimed at uncovering how biology and environment interact in mental health disorders. He recently received a Young Investigator grant to study the biological roots of BPD and continues to work toward improving our understanding of mental health through science that connects the brain, genes, and everyday experience. It is my pleasure to welcome you here today.
Thank you. So I just get started right away with the presentation?
Yes.
Okay, perfect. So thank you very much for the introduction. I'm trying to share my screen now. Okay, I think this still has that little top and bottom. I'll see if I can get it to complete full screen. I think I need feedback if you see the...
We see the notes. The other way was better.
The notes, okay. I think if I swap it now,
Yep,
now should
that's
be
perfect.
perfect. And now you don't have any additional things on the display. So yeah, thank you very much for inviting me to speak. This is a very rewarding interaction we had so far in the last month or years probably already. And I'm very, very grateful for the number of people who are interested in our research. So I'm a bit nervous to present because I don't think I ever presented in front of so many people. So yeah, my talk today is called Unraveling the Genetics of Borderline Personality Disorder. And as mentioned, I worked in the Central Institute at Mannheim, and this institute connected to the University of Heidelberg. I'm a psychologist by training, and I have worked mainly in the field of psychiatric genetics in the last years, now moving into the field of artificial intelligence applied to large-scale data in psychiatry. So I have no conflicts to declare. So I will give a brief overview of what I will talk about today. I will give a brief overview about psychiatric genetics. So some core concepts that are interesting or important if we talk about genetics, heritability and how we study it. More important, even I think the disorder model, what do we think is the genetic model or how's the genetics working that's underlying a disorder risk? And then what are genome-wide association studies and how do we run those and what are the results we can get from those. And this will be also the core of my presentation. So I will present one of these genome-wide association studies or GWAS, and this is the first large-scale study of this type for borderline personality disorder. And in the end, I will talk a bit briefly about ongoing projects that study BPD also from a genomic perspective and will lay out some future plans. And maybe you have some more suggestions what you think we should actually study. So when we talk about psychiatric genetics, one thing that's often a thing we need to argue, why do we even put effort in the subjects? Why don't we study other disorders? And one thing that's important is there's just a lot of burden of disease linked to neuropsychiatric diseases. And that's actually larger than for most other disorders or for somatic disorders. So we can ask, why do we need to investigate mental disorders? And I assume everyone who is attending has the opinion it's an important thing to do. Actually, what we can summarize, we actually don't know too much why and how mental disorders arise. We have relatively limited possibilities to identify persons at risk of developing a disease and also are quite limited to predict how a disease will develop. Treatment often occurs based on trial and error. This is also true for medication. So we just try the first medication. If it doesn't work, we switch to the next and to the next. And of course, it would be great if we could identify the right medication for each individual if a medication is needed. And from the biological perspective, we lack biomarkers, so biological measures that would inform us in any of those questions. So the aims to study these disorders from a biological or genetic perspective are we want to characterize the risk factors, we want to learn more about the risk factors, and we want to understand the biology. And of course, we also want to use this knowledge to predict certain disease courses or risk. And in the long term, of course, we want to produce research that benefits prevention and treatment. This is already highlighted in red here, and I think this is to say I don't want to overpromise on that. This is still a long way to go. This is not an easy task. We need a lot of big data sets. I will lay out some of those aspects later on. But I think it's important also if we do science that we don't overpromise, that we don't promise anything we can't keep in the end. So when we talk about psychiatric genetics, one important concept is heritability. And under heritability, we understand that a part of the variation that we observe in a particular trait or in the vulnerability for disorder can be attributed to genetic factors. And the heritability corresponds to the share or the proportion of this variation that we can attribute to inherited genetic factors. We can estimate this. Normally, this is done with twin or family studies, so where we know what degree of environment and what degree of genetic influences is shared between different persons, and then we can measure disorders, but also traits, for example, height or personality features, and estimate to what degree genetic factors contribute to this. So this is an example. The black bars are monozygous twins, so twins that have 100% same genetic information. And the gray bars are for dizygotic twins, so twins who are as related as siblings, so share 50% of their genetic information. And what is plotted is the twin concordance. So what you can see here is how similar are those twins. And this is for somatic disorders. I think I can point here, but this holds also true for most psychiatric disorders. So you can basically say for all psychiatric disorders. So yeah, we see the pattern. If the people are genetically identical, they're also more similar in regard to mental health phenotypes. And with this study designs, we can also estimate then the proportion that's attributed, can be attributed to genetic factors. This is relatively bold, but I think also quite well established statements. So we can say all mental disorders have a genetic contribution. On the other hand, this is not absolute, this is not deterministic. So we can also say all mental disorders have an environmental contribution. And what you also can say, this really differs between different mental disorders. I will have a slide coming about this as well. What I always try to communicate as well, if you hear something is heritable to a certain degree, that's always an estimate. That's an estimate of a specific population, an age group, a historical period. This does not mean this is set in stone. This might be a different estimate in a different population, a different subgroup, and so on. And also, this is something we estimate in the population. You cannot say a certain disorder is 80% heritable in a person, for example. This is a short plot on the y-axis. So to the top, you see how heritable these disorders are, going from 1 to 0, 0 to 1, 1 being highly heritable, and the lifetime prevalence. So to the right, the disorders that are more common. And what we can say about mental disorders in general, the more common disorders tend to be a bit less heritable. And when we talk about borderline personality disorder, it is somewhere in the middle. So it is heritable, but also has a large environmental contribution. It is a common disorder, but not as common as, for example, major depressive disorder. And we have certain assumptions in our work, and we also feel right now that those are backed up pretty well by the data that has been generated in the last years. So often if you think about a genetic disorder, you might think about a monogenic disorder. That's the genetic disorders you might have learned about in school. So there's a certain mutation, it causes a certain disorder. So this is here indicated by this little genetic variation. So one variation leads to a certain disorder. In psychiatric disorders, we talk about polygenic disorders. So there is a big sum of variants that contribute to our risk, all adding up. But that also means that all those variants, especially if they are common, which is what I will present here in this present presentation and in our JIVA study. So if they're relatively common, those genetic variants, they will have very low effects. And we can compare them to genetic variants. For example, if we would look at height, they might add half of a centimeter to your height. So they will not make a huge difference. But of course, if you have a lot of variants that either are linked to high height or low height, this will influence how tall you will be in the end. And this is also a disorder model that we use or we assume. So this is a threshold liability model. So if we look at symptoms in the population, so on the y-axis you have the frequency and on the x-axis the number of symptoms, you will see most people have a low number of symptoms, so 0 to only a few. And then there is a tail in the distribution, some people that have more symptoms. And here I list the symptoms of borderline personality disorder based on the DSM-5. So normally you would require 5 of those 9 symptoms for a diagnosis. There's also other aspects to it, for example, suffering. But on the symptom level, you would need 5 of those symptoms to qualify for a diagnosis. And so only if you're over a certain threshold, we would talk about a diagnosis. So this is an example. Someone has fulfilled 5 symptoms, for example. So what we do out of this continuum, we will say some people are affected if they are over a certain threshold. And now if we talk about the genetics, we assume that underlie this, we think about a continuous risk. So we have a genetic risk for a certain disorder, and we assume most people have a pretty much average risk. So it's a bell curve. Most people are pretty close to the middle. And then there's a few people with a very low risk and a few people with very high risk. And we assume that this continuous risk contributes to your disorder risk in combination, of course, with environmental risk factors and so on. So this is the basic model that underlies our calculations. Now, how we use these ideas to study mental health is in the data I present, genome-wide association studies. So the genome consists of a sequence of different nucleotides. You see 2 examples here, and it is all pretty alike, but every now and then there's variation in here. And what we see here, there's 2 alleles, and people differ in those alleles. And while we have 3 billion base pairs in our genome, we assume that around 10 millions or more than 10 millions of this variance exist. So there's a lot of variation, but as I said before, it's just gradual variation. This doesn't make anyone sick or gives anyone a substantial huge risk, but it adds up. So this is our model. The highlighting is shifted. Of course, the 2 different alleles would be the 1A here versus the C that you can see here. So what we can measure with modern methods is we can measure those variants, and as I showed in the slide before, we distribute those evenly across the whole genome. So we don't sequence the whole genome because this is still very costly, but for quite affordable amounts of money, we can put a lot of markers over the whole genome and use this to analyze the genome. So what we have here is no prior hypothesis. So I told you a lot of assumptions we have, but we don't have a hypothesis what we will find exactly. And that means we're independent of any traditional beliefs, what is relevant for a disorder, but we try to find something new, something unexpected, to find some new input to our knowledge about the disorders. And what we will do here, we just study all these variants and we compare, is something more frequent in cases compared to controls. And we test this for this up to 10 million variants. And if one allele is more frequent than in the cases than in the controls, we will see this in a statistical test. And this is then plotted here. So the higher the dots are, the stronger our significant signal is. So this means it needs to be very significant until we say this is a real finding, because we do so many tests, we do very strict corrections. And those are called Manhattan plots. So this is basically depicting the whole genome on the x-axis, and then the significance, so how reliable is this result in the y-axis. And there has been substantial progress in the last 2 decades. So there's big consortia, the PGC, which I'm part of, there's large biobanks such as the UK Biobank. And what you see here, this plot, I don't know how easy this is to read. So we try to find significant loci, gene loci. This is the number of cases that went into a specific study. So the number of patients, as you could say. This is the number of significant genes or gene loci we identified. And as I said before, we need very large samples for our research, and this is shown here quite well. And it differs a bit between different studies or different disorders. So schizophrenia is the poster child of psychiatric genetics. Maybe it's the first disorders where a lot of loci were identified. But also here, we needed over 40, 50,000 subjects to come to the current numbers. In depression, which is less heritable and more common, there needs to be larger samples to find the same number of loci, even though the newest results that are not on this plot here, which is already 4 years old, also show up to 600 loci or more than 600 loci we identify, and also bipolar disorder caught up. But you can see the numbers of cases included in those studies. For depression, more than 300 cases with depression are included here. So where are we with borderline, or where did we start with borderline? Actually, it's interesting. This is a bit understudied, and it's surprising because it's not so rare, and it's actually even more common in a hospital setting where physicians might study disorders. It has been established that it has a genetic influence, and actually it's quite comorbid with other psychiatric disorders. It shares clinical symptoms also with psychiatric disorders, at least to a degree. So one could assume this should also work similarly for genetic studies, but it is really under studies. There's some twin studies, of course, of it, and they have really interesting results. But from this molecular perspective, where we really try to find the genetic variants, there is not too much that has been done. So the biological underpinnings are largely unknown. As I also said before, it's under-researched, so GWAS might be a good way to gain insight. And so we did the first study with just 1,000 German cases, and we just got there. So very small, no identified loci. We had some signals in the results that told us this is leading somewhere, but it was still way too small. So this is some people that work with me on that. This is a big team effort. I'm just one of the persons who is invested in it, but it's a big team of people who supported that or who also drove that. So it's Marcella Rietschel, my old supervisor, Stephanie Witt, Alicia Hall, who did this on the Mannheim side with me. Alicia now moved to Denmark, and Swapnil and Stefan from Charité Berlin, who are really heavily involved in the statistical part mainly. So we had this German borderline consortium that was this data I just showed, and we decided to go as international as we could to proceed with our study and to get bigger samples. So this is a map of the countries we could get to participate in our now called International Borderline Genomics Consortium. And this is more a question for the students if I show this. We see that these countries are all Western countries. So this is something that is an issue for us. I need to make a very brief break. 1 second. Okay, sorry for the interruption. So this is an issue we have with genetics. We just get easy access, or data is available largely in Western countries. We study largely people of European ancestry. And this is something we would like to change, and I hope we'll be able to change in the future. So our aims were to identify genetic risk loci of BPD, and by that to learn more about the risks underlying the disorder. So this is our study design. I think it's maybe a bit difficult to take in all the information. So there was a lot of small studies that contributed, 17 studies where people had collected data, blood or DNA generated already, or we generated it. But we also had big studies from those biobanks I mentioned before, where people contributed results that they sent to us, and they investigated the association of borderline and the genetic factors. So in total, that was a discovery analysis of more than 12,000 cases, more than 1 million controls. And we did the replication analysis in more than 600 cases to see if our results could be identified there as well. So if we look at the results now, what we can see here is that this is a scheme that describes what can we do with these results. So we generate this association findings that basically say what genetic variants are associated with the case control status of borderline personality disorder. The first thing is we can look at the single variant associations, and we can also say something if the heritability we find in the twin studies or family studies is also observable here. So what we see first, we do identify now for the first time a genetic loci of borderline personality disorder. And we found 6 independent loci in the primary analysis. And when we analyzed it together with our replication sample, this number increased to 11 loci. And if we estimate from our results how heritable this is, we have an estimate of 17%. So this is lower, but that is something that's commonly observed. It's lower than the number from twin studies, but we also have the contribution from rarer variants, for example, that we don't study here. So this brings us here now. So we are getting somewhere. We're getting some results. We identify some genetic loci with some genes. We collected a few more samples, but it's still quite behind other disorders. So what we find if we look at this loci, we can look what gene is right there or right next to this region we identified. And for example, this is FOXP2, is a very interesting gene. It's relevant for neurodevelopment, for language development. So this is our biggest hit in the analysis. But also FOXP1, that's also related to it. But then also some other disorders and some other genes implicated. And for some of them, we can find associations with other mental health disorders or with measures of personality. For others, they're more linked to metabolism, for example, or other disorders. What we did was to query drug-gene interaction databases to see if we have some evidence that certain genes indicate that certain medication might fit onto or on top of those biological systems we identify with our GWAS analysis. And for example, for one of the genes, we see that lithium pops up of the gene that has been associated before. So I highlight this here, but I also want to caution, I think we are far from where we can really make any certain conclusions with our sample sizes. This is also only a probabilistic thing, even in the larger GWASs. This will make a trial on a certain medication more probable to work, but it's not a guarantee just because we find genes or a pattern of genes that maps a certain medication. This can never guarantee us that a medication is actually helpful for a condition, but it might increase the probability and might guide where new developments are invested in. So I already mentioned the FOXP2 gene. This is a very interesting gene. And what I also found interesting to see that this has been implied in several other genetic studies that didn't study borderline personality disorder, but other relevant phenotypes, for example, maltreatment, cannabis use disorder, antisocial behavior, and PTSD. And there is one of the top genes in each of those. So what else can we do with these results? And this is just a relatively narrow zoom in what we can do with these results. We can do way more, of course, with a lot of bioinformatics we can make use of. So again, this is if we do a gene-based analysis, you see some of the genes that were identified before also show up here. And this is integrating the SNPs from a certain region defined by the genes and where they are located. So what I just wanted to highlight here is this: we identified the gene also on the X chromosome. It used to be an issue that people ignored the X chromosome just because it is more difficult to analyze. Females have 2 copies, males have 1 copy. But of course, it can be highly relevant, and it might be especially interesting to see also because we observe different patterns of gender distribution in the diagnoses. So what else can we do? We can also overlay this data with data of gene expression. So what genes are active, for example, in certain tissues, in different brain tissues, in different cell types, to infer where these genetic variants might be actually acting or where they might be relevant. Because we cannot easily look into the brain, it is, of course, very interesting for us to do this via bioinformatics. So what we see basically here, our results are enriched for brain tissue, so the genes that we find are relevant in the brain in certain cell types. And also, if we go to developmental phases, we can see that certain developmental phases are especially relevant. And this is here, for example, early prenatal phases that are highlighted here. So this is also all limited, but we can already have a certain insight into relevant processes or tissues. And it is the case that the bigger our data gets, both this tissue data sets that we connect to our GWAS, but also our GWAS, the better our, or the lower the margin of error will be, and the more certain we can identify relevant processes. So what else can we say with it? We can say how closely is this genetic structure that we identify here related to the genetics related to other disorders or traits. So this is a background slide. So there was a big study that tested this systematically. And basically what we can use the results from our GWAS studies and see how closely linked are they. Are the signals in the same direction or in the opposite direction? And if disorders show a very similar pattern, we can assume they are closely related biologically as well and in regard to their risk factors. So what we see here in the scheme, while neurological disorders, this study disorders of the brain, neurological disorders are pretty distinct genetically. There's a lot of relatedness between mental health phenotypes or psychiatric disorders. As you can see here, also some disorders are very close. This is the dark bars here. For example, anxiety and depression or PTSD show very close links, or bipolar disorder and schizophrenia, while others are more remote here and less closely related and similar. So when we look at these approaches, we call it genetic correlation. So basically, +1 would be a perfect match, fully corresponding in a positive way. -1 would be an opposite signal. What we see here is that we see a very broad range of positive genetic correlations, mental disorders, but very high correlations with PTSD, depression, ADHD, and psychiatry, but also a history of suicide attempt. This is all other GWAS someone made the effort to generate, and we could download their results and correlate them with ours. So it's correlated with self-harm, suicide, but also personality features, even though less strongly. And what we thought was interesting, also in a positive way, so shared genetic risk with diabetes, for example, asthma. So we can use this data that we generated, and we will make them publicly available for the community. Everyone then can use them to look at shared genetic signals, for example. And one technique that's also a very common thing that you will see a lot in the literature and that's very commonly used is polygenic scores. These can be compared to other scores. Often we call them polygenic risk scores as well. For example, there's risk scores for cardiovascular disorders. And here you will add up the age group, the gender, cholesterol levels, blood pressure, diabetes history, smoking history. You will add up these risk factors and you will end up in a risk score per person. So some people have none of those risk factors, so they have a risk score of 0 for this cardiovascular score. And some people have nearly all of them, so they have a close to maximum score. And then a lot of people will have middle average scores. And this is what we can do with our genetic data. In our GWAS, we identify, is this a risk variant? Is it a protective variant? And if we have other data sets where we also have genetic information, we can calculate a risk score in this data set. So this is shown here a bit. So we have a genetic risk score that goes from low to high. And we see normally that this is on average, but as well not deterministic, higher in patients than in the controls. You see this is overlapping, but there's more and more people in the patient group that have high risk scores. And we can use those as a tool to do research. And what we did here is we divided the risk scores in bins. We started, so the lowest 10% of the score, the next 10% of the score distribution. And so what you find here is the 10% with the lowest risk score. This is the 10% with the highest risk score. And if you focus on the blue dots here, what you see is the odds ratio, so kind of how much is the risk increased. And you see, compared to this lowest group, the higher your risk score gets for our disorder, the higher your probability to be a case of this disorder also gets. So this is a genetic predictor, you could say, integrated in one measure. And this is just basically to check, does our GWAS work? We predict borderline in one sample with the risk we identified in the combined analysis of all the other samples, and it tells us something, how much risk have we already identified. And this is also something that will get stronger if we increase our sample sizes. If you get bigger studies, then our prediction will improve there. The blue dots are the results based on our last analysis, where we just had this 1,000 samples. So you see that we basically couldn't get much prediction out of there, but now it really makes a difference. Still, this just explains a few percent of your variability. So it's not too informative, especially not on an individual level, but it's a very valuable research tool already. So how can we use this? One approach is phenome-wide association studies. So from the genome-wide, we move to the phenome-wide. Phenome is everything we can observe. And this was done in a hospital biobank. So we sent them our results so they know what is the risk variant, and they calculated these risk scores I just described in their hospital data set. So they have more than 60,000 individuals, and they tested the association of the risk of the borderline risk genetic that they could identify based on our results. They tested what disorders actually associated with it. So what is linked to that? And what we see here, this is again a bit like those Manhattan plots. So there's different disorder groups. The red line is everything that's higher than that. We think that's statistically reliable. So that's after correction for all those tests we do over all those disorders. What we see here, primarily mental health conditions are linked to the genetic risk of borderline, as we would expect. And this is all in a positive way. So it's a positive association. And that's mood disorders, anxiety, but also substance use, PTSD. So it's a broad range of different mental disorders or related phenotypes, suicidal ideation or attempt, for example. But we also see, again, some somatic disorders, for example, chronic airway obstruction or obesity shows up here. So there's some link between this condition, seemingly on a genetic level. Now it's
Perfect.
good?
Yep, perfect.
Okay. So as we probably also lost some more time. So this is basically the same study, just this time we didn't share the data with the hospital sample, but with the UK biobank, so a population cohort in the UK. And they basically did the same. They looked what our risk score, so the genetic risk for BPD that's identified. There's a lot of rest of it that's not identified because our study is pretty small relatively. And again, it is linked primarily to mental health conditions, but also to a lot of other aspects. And of course, that's a very interesting follow-up thing to study. How do those links work? Do they work via behavior, maybe via immune pathways, and so on? But we couldn't follow all the questions that our results raised, of course. So this is the summary of this GWAS data. So we identified first risk loci for borderline, and it highlights really interesting genes that, of course, could also be investigated now in a lot of detail. We see basically it behaves like most other mental disorders. It has a strong genetic component, but this is very distributed throughout the genome. It is very polygenic, we call it. So it's not one or two genes that are super relevant and the rest plays no role. What we identified, those 6 to 11 genes, this is the peak of the iceberg. we know from our experience, if we increase our data set, we will find way more linked genes. Our enrichment analysis tells us there's brain tissue highlighted, so the genes that we identify are probably relevant in the brain, which fits to our perspective of mental health disorders as brain disorders. There's some discussion about that, but I think can be said like this. It highlights early developmental phases, but also here, the bigger would be the better. It would be better if we have more reliable data. It shares a very wide genetic risk profile with other mental disorders and traits, but we also see some associations to somatic health, and it would be really, really interesting to follow up on that in more detail. And what we did is, I think, a bit of a showcase. What can we do with these results? But this is available to the community. We have shared this already, and with the publication of the manuscript, it will be more freely available. So it can be and will be used in a lot of studies that just try to target what is the etiology of BPD and how can we use molecular genetic approaches for this. So I put two barcodes here, but I already said before I can also share slides after. So this is a group we host. It's mainly linking researchers interested in those disorders in a genomic way. And this is our preprint, so it's not officially published yet, but it's already available for reading. Exactly, okay. So just now as the outlook, what are we currently doing? This is our main publication now, but we're doing a lot of works with those data sets or with other aspects of BPD or with other genomics that we can measure. So we also share data with the PGC suicide working group. They look at suicide ideation, attempt, and also completed suicide. And we have some information in patients if they attempted suicide, so we can share this information or the genetic results of this analysis with them. We contribute also to analyses that look at personality traits, so not in a disorder perspective, but just a variation in the population. And we try to look at other omics. That means, for example, epigenetics, gene expression data in postmortem tissue. That's the NARSAT grant I was funded. And we look at shared genetics. We have some projects that started. One very interesting also are traits that we measure in the general population that are like borderline traits, borderline personality features. Are they actually linked to what we see here in this case control approach? That is a very simplistic approach to just put people in a case control group and see what is the association. So future plans, and I think that's already my last slide with some content. So we want to continue this. I think this is a very worthwhile effort. We need to be larger. So we try to motivate people who have data to contribute. This will be easier now when the paper is published. That motivates people to be part of the next one. Of course, we are also looking for funding to be able to have manpower in this analysis. I stressed we need to be more diverse, different ancestries. I hope we'll be successful in that regard. We have this multi-omics analysis, also looking at proteomics. And in our group, we try to use also AI method to combine those risk scores of several disorders to get an improvement in prediction and also identification of meaningful biological subgroups. So I have a last question that just says, what would you like to see investigated? I'm really, really grateful for this opportunity to present in front of all of you. I hope I managed to present this in a way that this was informative. I think we'll be open for questions now. So thank you all very much for showing this interest in our research. And when I say our, I showed a few pictures of some core persons that are involved in this. But I think this is the full authorship list from our paper, and everyone did something. and there's probably a lot of people who collected data, interviewed participants, who prepared data, and so on, that are not mentioned here. So this is really a gigantic effort. I think I can only thank everyone, and I need to thank the participants of our research, and of course also founders of our research. And with that, I'm done. Thank you so much.
Thank you so much. This was a lot of great information, and I don't know if you can see the emojis all going
I
through,
will
but it looks like a lot
probably
of people.
stop my screen.
Oh,
Oh, thank
I
you so
stopped.
much.
I
This
stopped.
is
Yeah.
amazing. Thank you so
Yeah.
much.
Yeah. So we do have a couple of questions. We'll try and get to as many as we can. Please put your questions in the Q&A if you do have one. We have a couple people who've asked how they could participate in studies like this.
Yeah, this is a good question. So we don't have an ongoing recruitment program or something. I know we have discussed this. In principle, one just needs to get some information and some genetic material to someone who has the capacity to study it.
Yes.
Um,
All right. I'm looking through the questions here. We do have we have a bunch. Um, so we have a question here asking for are there any significant epigenetic factors that you've identified in BPD?
so the samples, that's something we learned. So there's some studies on epigenetic factors. And there's some results, but I think there also we learned over the years you also need to combine data, and you need to generate big data sets. It's a bit the same, also the easy but compelling stories with one gene being changed through some environmental experiences, and then this mediates your well-being. This doesn't hold true so well. So we generated some data. We are not incredibly impressed so far, but it's also still a bit fresh. So, but we will reach out to also everyone we are aware of that has data. We, I think, connected to everyone who generated data, and it's one of our plans to also do a meta-analysis of that. But yes, it's a lot of work, and we need to balance a bit what we focus on. So this, what I presented today, was one of our biggest focus. Then this brain analysis with the NASA grant, another one. But it's definitely very interesting in regard to the disorder, with a big focus on trauma as well, to highlight that more, and maybe also how this interacts with genetic factors. So a lot of work to be done still. Yeah.
Yes, thank you. Um, so our next question, let's see, we have about 70 questions popping up. Do you have enough data to, I know this question was somewhere up here. Do you have enough data to do some subgroups to look at, based on different ages, different factors, analysis, disease severity, prognosis?
So we did by sex analysis, for example, in our manuscript, and that looked relatively correlated. So there's a strong overlap of the factors influencing it. But for example, here we wouldn't have the power to really detect differences. That would be require especially more male participants. One issue we have with some of the data, it's a bit of a technical aspect. We need to have the information also to have information on the subgroup. So as I showed in the beginning, we have a lot of studies that go in there. So we need to really sit someone down to curate this data, and then the sources are really different. So it's a lot of, creates a lot of work, but I would like to look more into that. And we compiled some data on depression, on suicide attempts. So we will look at that in future analysis if we see any differences there.
Thank you. And then we have a couple of questions about some diverse populations. It made it seem like there was a lot of European descent, and people are asking, from other countries whether there's, African descent represented, Middle Eastern, or from India.
Yeah, yeah. No, I stressed that before when I showed the world map, and um, it's a pragmatic thing we did. We just included every sample we could get our hands on, basically. So, and that means those samples already existed normally, and they had collected material and could generate genotyping and so on. And but what we have included in the analysis is only European in the moment. We are aware of some data sets that are mainly American or US American, where there is then subgroups that are big enough of African or Asian descent, for example, that could make a own group, because it's also not trivial to just jointly analyze it to avoid any false results. So it's something we need to also develop the field more, how to either jointly analyze or how to group data sets and so on. We did some more of these efforts in the last depression study, but there we just have way bigger data sets. This is, I think, more than 600, yes, nearly like 50 times larger. So, and there we were able to increase that share. But it's definitely a challenge for the future, and I hope we progress there a bit.
Thank you for sharing that. What we'll do with a list of questions that are keep coming in is we'll review them as a staff. And Dr. Streit, if we can come back to you with some of the core questions, and maybe we can send it out in an email to those participating, because we do have a lot that are coming in. And I do want to thank you, thank everyone who joined us. I think we had close to 700 people on, so people were definitely interested in this topic. And we thank you for engaging with us and joining us today. And just a reminder to the audience, we do have an upcoming program called our Chat BPD. It's an event where people with lived experience talk about different topics. Our November one is scheduled for November 30th, and the topic is self-acceptance. So please be on the lookout for an email about that event. And we thank Dr. Streit, we thank you for being here with us today, and we thank all of the participants who are on.